Good morning, all. I welcome all the participants and all the members of the organizing committee to this morning session. Today, we have with us Dr. S. Geeta. Ma'am is uh, Director, Center for Plant Breeding and Genetics, Tamil Nadu Agriculture University, Coimbatore. And the topic of discussion is recent advances in breeding for biotic and abiotic stress tolerance in sugarcane. Ma'am, you are visible and audible. Also, your presentation is visible. Please start, ma'am.
Okay, thank you. Thank you for the nice presentation. And first of all, I would like to thank the organizers for giving me this opportunity to present a lecture on recent advances in breeding for biotic and abiotic stress tolerance in sugarcane. Because this particular crop is very close to my heart, so I'm very happy to present whatever I know in this particular field, especially in abiotic stress tolerance breeding. So outline of my presentation would be, first I will be giving a brief introduction, why it is required, and going to abiotic stress tolerance breeding. So in that I will be covering screening techniques, mechanism of tolerance or resistance, genes involved in conferring the tolerance, and utilization of wild relatives, and also breeding methods, and what is the possibility for the mass using mass, and just about this tolerant varieties under cultivation, and also giving a brief information about this donors. So similarly for biotic stress tolerance also. So under abiotic tolerance, I have planned to cover drought, salinity and water logging. Even though high temperature and damage due to low temperature is there, I have taken these three major abiotic stresses for this presentation. And in biotic stress, I'll be covering about red rot, smut and borers. Even though there are some more viral diseases are also there, so I'll be concentrating on these three pest and diseases, and possible improvements in selection methods to achieve a higher level of resistance or tolerance for this biotic as well as abiotic stresses. And I'll be concluding with some way forward notes. So before going into details about the breeding methods, so all of us know, see, if you see this particular picture, almost all the states in India are vulnerable to one or two or even three types of stresses. When you take into account of southern states, it is vulnerable for both extreme level of stresses, both drought as well as waterlogging, and some of the North Indian states are vulnerable for drought as well as for high temperature. So sugarcane is cultivated almost in all the states. So there is a dire need to develop varieties to cope up with all these stresses. And you can see almost all the districts in India, some of the red color districts are in more danger states because of this climate change. Water depletion is there and high temperature is there. And not only the drought and waterlogging or cold temperature, we have the problem of problem soils, that is saline soil, sodic soil, and it is increasing in the area because of over exploitation of groundwater and use of higher level of fertilizer, what more than what is required. And it is projected now, it is 67 lakhs hectares under problem soil in India, but it is projected by 2030 it will be doubled, almost double. Almost 11.25 million hectare is expected to be affected by salinity or alkalinity. And besides these abiotic stresses, because of this climate change, we frequently get the problem due to new pests and diseases, and also some of the places where hitherto no occurrence of particular pest or diseases, we are seeing that minor pests have become major pests. So because of this climate change, so much of stress is given to crops as well as the farmers, and also as well as for the breeders who are involved in the development of stress tolerant crop varieties. So then how we are going to achieve the sustainable agricultural production under all these problems, abiotic and biotic stresses? No fears. So, see, of course, by 2050, we have to almost double the production, agricultural production, 2.5 times we have to increase, but under the land pressure. See, so far in till 1960 or 1980, 1 hectare was sufficient to feed uh, uh, three people. Now the same 1 hectare has to provide the food or other agriculture commodities for the 9 people. So because of this population growth, one side population growth is there, other side land under cultivation is also decreasing. So we have a pressure on land for cultivation and reduce losses due to storage as well as pest and diseases. And so under these circumstances, under these limitations, how we are going to achieve 250% increase in agriculture production? Of course, with good management practices, and very big help is going to come from the breeders and biotechnologists. 80% contribution is going to be done by the breeders and biotechnologists. So containing the crop losses due to various environmental stresses is a major area of concern to cope up with increasing agricultural production. So definitely we have to, there is no escape, we have to develop climate smart varieties to achieve climate resilience and sustainable agricultural production. So when we look into the breeding strategies in sugarcane, so from simple breeding to modern breeding till 2020, so I'm not going to touch upon these things. So hybrid breeding, sugarcane is an excellent crop where we have completely exploiting this hybrid breeding. So I have given 5 stars. And similarly, tissue culture, it's a very good crop which shows good response for the somoclonal variation or in vitro mutagenesis. But coming to mass, so mass, we are lagging because of this complexity of genome. Still, we have not gone to the level of other crops like rice, wheat, and maize. And similarly, mutation breeding, we have done a good work. Breeders have done a good work and come out with many mutants with economic importance. And coming to the recurrent selection also, even though it is not that much successful or followed in other crops like rice and other self-pollinated crop, even in maize it is not practiced to that much extent, but in sugarcane it is easy to follow also. We are making so many number of intercrosses in ISH, between ISH, we are making so many crosses, and we are doing our level best as far as this recurrent selection. And GMOs, it's a very good crop for transgenics. So much of work is done on development of transgenic sugarcane, but to my knowledge, so far no transgenic variety has been released. And genomic selection also to capture the non-additive genetic variants is an excellent tool. So it is also being practiced. So I have given 3 stars. And speed breeding, finally I'm coming to speed breeding. So it's in the infantry stage, I can't give any star for that. We have to go a long way for this speed breeding. So all these stresses, drought, waterlogging stress, so they give trouble in growth and development of the crop and alters the sugar content or sugar recovery and yield. So finally loss in sugarcane yield as well as in sugar productivity. So in drought, again, I'm showing all the sugarcane growing areas or states are vulnerable for drought. This picture very clearly shows. So this is the primary abiotic stress occurring in almost all cane growing areas of India. So drought reduces sugarcane yield up to the level of 60, in severe conditions even 90%. And luckily we have some good donors, proven donors for developing drought tolerant varieties. Even though they are all old varieties, they are all proven donors for importing drought tolerance in sugarcane varieties. So one of the selection criteria which breeders fail to do is deep root system, because it requires some more exercise, also rhizoctron facility and other things. So what I feel is if we concentrate on this below ground parameter like a deep root system, I think we can make some more progress in developing drought tolerant varieties. And a drought in early and mid growth stages mainly reduces cane yield, leading to low sucrose yield. But a moderate drought in late growth stage can improve sucrose content because it facilitates the senescence and good sugar recovery is there. And so coming to the, so this formative phase is the very sensitive stage for a drought. So we have to develop varieties, sugarcane varieties that tolerates the drought at the formative stage. That is what very important here. So both ABA dependent mechanism and ABA independent mechanism operates for conferring drought tolerance, even this osmotic adjustment. So at the level of germination and the early stages growth, this osmotic adjustment plays a major role. And during the growth, for subsequent growth, stomatal closure plays a major role. So so other things are ROS scavenging mechanism that is done by some of the genes, other secondary metabolites that takes care of this ROS scavenging. So other thing is ABA dependent mechanism, which takes care of redox status as well as ethylene response. So at we have one research station at Melalatur in Vellore districts of Tamil Nadu, which is doing an excellent work on developing sugarcane varieties for drought tolerance. What we do is, so we have the field like this. So we just keep some 6-meter gap in between two fields. So this will be irrigated. Okay, so normal irrigation will be given. So here we withhold the irrigation in the formative stage, especially even though formative stage starts from the 60th day. So the critical stage, as per our observation, starts from the 90th day. So we withhold the irrigation from the 90th day to 150th day. So we raise the clones here, either in 3 rows or 4 rows in the replicated model, and we screen the drought tolerant clones. That has given us good results also. So, and we can see the leaf rolling is one of the mechanism that confers that drought tolerance. You can see. And there are some biomarkers also for drought tolerance. One, why I am mentioning here is lipid peroxidase or ethylene. No, so even though limitations are there in using the isozymes for identifying the drought tolerance level, now that we have to take up these markers, isozyme markers, biochemical markers, and identify the candidate genes so that we can identify the markers, DNA markers closer to this candidate genes. Now that the genome sequencing facility is in a big way, so it is quite, it has become little bit cheaper, relatively cheaper. So, these are all the main candidate genes, especially this glycine, betaine, lipid peroxidation, and prolines. So we have to identify the DNA markers related to this or closer to these candidate genes and go for the selection at the early stage itself. And again, you can see you'll be wondering differential gene expression, and see all for this drought tolerance. See, all these genotypes have been reported to be drought tolerant. But you can see the number of genes expressed in various studies. So it varies. It starts from 93 genes to 1,670 genes. So another thing is, it is genotypic, not only genotypic specific, as well as growth development specific also. If you say the same genotype SP90 1638 and SP19 6038 here, so when it is subjected to drought at the 90th day, 93 genes are expressed, whereas when it is subjected to at the 60th day, 432 genes are expressed. So it's very interesting. So complex defensive mechanism are activated in response to water stress. So under this very complicated situation, how we can reduce the error, how we can come to the conclusion of drought tolerance. So what I would suggest is, we can raise the crop. See, we can shortlist at one location and take the same entries to the other location where a kind of drought is prevailing. So by doing multi-location tests, I think we can confirm the drought tolerance in the particular drought tolerant clone. So because we don't have strong markers, molecular markers for the drought tolerance. And another best technique for developing sugarcane drought tolerant varieties is that is in vitro mutagenesis and using the peg. So as I already told, sugarcane is a very good crop for doing excellent tissue culture work or creating somoclonal variation. So with this work, what in this particular study, what they have concluded is peg at 2800 level, no, that is optimum for creating the variability. So this picture clearly shows, see here and all at this level, you can see 16%, you see more number of plants, but this is 24% seems to be good, only very few plants are there. So that would be more reliable also, that might give you in the field also with more level of drought tolerance. So at 6000, 32%, you see none of the plants was surviving. So this 24% has been reported to be optimum. So, and we have the support from our wild relatives also. All the Saccharum complex wild relatives are giving tolerance for one or other biotic or abiotic stresses. Like miscanthus is giving tolerance, miscanthus, spontaneum, barberry, erianthus are giving tolerance. They have some genes for cold tolerance, and again they have the genes for the drought tolerance, and also for salinity, and robustum is having some good genes for waterlogging also. So, and Erianthus, a potential donor for drought tolerance, but only problem here is you have to synchronize the flowering by altering the photoperiod length artificially. So that is the only problem that we have to ensure. And this is the work from China. They have come out with what they claim is this particular line with Erianthus arundinaceus is the best. They call it as a high class drought tolerant line, Heng 95-41. And in from SBI also they have come out with good lines in progress with the Erianthus genome, that is GU0428 EO02, lowest drought susceptibility index. That means it has got the highest level of drought tolerance. They show 93.9% drought tolerance line. So this can be, I would advise, this can be used in hybridization program wherever drought is the major problem. Again, another interesting information I could get is not only when we in progress this or when we cross this Erianthus with a Fishnerium or a Spontaneum, much of cytoplasmic genetic content also transfer to the hybrids. So we have to explore maybe those genome from the cytoplasm from the of the Erianthus might contribute for some of the specific biotic or abiotic tolerance. And we are fortunate that we can identify the introgression of this Erianthus genome with the help of 5S rDNA, ribosomal DNA markers. So without this, we could not have achieved the introgression of Erianthus. This is one of the hybrids in one of the literatures I could find. So this all this green or Erianthus genome with the GISH analysis, they have shown clearly in some of the hybrids only one chromosome has been integrated, and some of the lines even 12 chromosomes from Erianthus has been has gained the entry into this hybrid. And again, Dr. Mohan Raj has published another article. So he has shown this Erianthus procerus, not only the arundinaceous, even Erianthus procerus could be the potential donor for conferring drought as well as red rot resistance. And you can see some of the lines with Erianthus genome in progress. See here, it is showing high level of drought tolerance here, along with red rot resistance. And another very rare hybrid they have got from China, that is Erianthus with Naranga. So this cross compatibility is very very low, but still they were able to get one or two seedlings with this combination, Erianthus and Naranga, and they are doing so they are further taking up this hybrid for other studies. With that, how they have confirmed this hybrid is the molecular primers of MSS IR66, SMC 72 720BS, and SMC 597CS. With these molecular primers, they have confirmed the hybridity. So any material coming out of this, or any material, if at all it is used as a pre-breeding material, we can expect a novel recombinants for many of the biotic stresses, tolerance for many of the biotic and abiotic stresses. And a very few reports I could see as far as this DNA markers are concerned. See, SSR markers are user friendly, especially for the breeders. So I could find only one paper, maybe I might have missed some of the papers, especially for the dehydrin genes. So dehydrin genes are involved in conferring, it's one of the candidate gene that produces some metabolites, secondary metabolites that helps in the adjustment of osmolysis. So in this, no, dehydrin proteins with the work from I think it is from Egypt. They have reported some of the SSR markers for this dehydrin protein, and they have shown these markers are reliable for discriminating the drought tolerant plants. So these dehydrin genes have been proven to be conferring for drought tolerance even in wheat. So, so we can also try these markers for discriminating the drought tolerance. And again, some of the markers, from Thailand, they have reported some SSR markers, so which I have listed here, SSR 9, 18, 19, 70. These SSR markers also help in discriminating the drought tolerance lines, which we can. So these are involved in ABA signaling pathway and hydrogen peroxide scavenging, peroxidase ROS scavenging mechanism. So this is also another information which we can also try. And validation of identified drought response candidate genes has been done by Hemaprabha at SBA, and they have come out with these genes, ABF, this is calcium signaling pathway, and this one is leucine related transcription factor binding sites, and LEA, we know, late embryogenesis abundance gene, and these are all the gene, again, this is taking care of the energy sensing and stress adaptive mechanisms. So they have validated these genes are definitely confirming drought tolerance. Now that we have to find the DNA markers with with the help of the genomic sequence and the bioinformatic tools. So they have very well confirmed these genes are responsible for conferring drought tolerance. And the clone SES16B and IRIT2, this is spontaneum, and they say these are all the repositories for the all these genes, drought conferring genes. And another work is AT DREB gene. So DREB is also known for its drought tolerant even in rice, for the terminal drought, they claim this DREB gene is very much helpful. So this is also they have identified the promoter that induces drought tolerance. And this is overexpression of sugarcane MYB. See, in the previous slides also we have seen this MYB. So this is another confirmation. This MYB gene helps in, this is actually involved in synthesis of antioxidants. So this transcription factor deciphering tolerance through drought and salinity, this is once again confirmed. And coming to salinity or alkalinity or sodicity tolerance, so the sensitive stages are germination, tillering, and cane formation. So when the EC is more than 2, we can expect 10% cane yield reduction. See, this is a glycophyte. When the EC is 3 to 6, 25% reduction, and when it exceeds 50, we can we can see 90% cane yield reduction. Similarly, under sodicity condition, when it exceeds 20, no, 50% yield reduction. And it actually the sugarcane requires only optimum ESP of 15 only. When it exceeds exchangeable sodium percent exceeds 15%, we can see for every one unit increase of this 1% increase of sodium percent, we can see 10% reduction in cane yield. However, we have got one salt tolerant variety Co 85007, which can be used as a donor for developing salt tolerant varieties. Similarly, we have also one from TNU, we have got one salt tolerant variety CoG 7 as well as CoC 26. And genes for drought. So this double trouble double trouble, I can say this is drought and salt tolerance. There, of course, this using this calcium signal. No, calcium is playing a major role in signaling pathways. So the calcium related genes, this SSCBL1 and SSCBL6, these two genes have been reported to be conferring for tolerance for both the troubleshooters like drought and salinity. So this it is better to screen the sugarcane for salinity or sodicity using the microplots. We have to create different gradients of salinity or sodicity, and under which we have to screen our material. So these are all some of the symptoms. Whenever there is a high level of salinity and sodicity, there is a problem in getting the micro or extracting the micronutrient from the soil for the sugarcane crop. You can see the symptoms of so whenever you identify the saline tolerant and sodicity tolerant, in general, you can conclude they are able to manage the, or they are micronutrient efficient lines also, especially for zinc or iron or boron. So if they are performing very well without showing any symptom for boron deficiency or zinc deficiency or iron deficiency, in general, we can conclude these salt tolerant or sodicity tolerant lines are efficient micronutrient efficient lines. And in another study by Priyanka, I could see they have compared many lines under normal and salinity, and you can see under salinity specifically some of the proteins, they are expressed at a high level. So specific proteins with high molecular weight or expressed under salinity. So now our job is to elude these biomarkers and go for the sequencing and to identify the markers, DNA markers. And so these varieties BO91 1158 and 92423 have already shown resistant against salinity stress. So you can see these lines here. Okay, I could not see these lines here. 11580. So this is the information. So wherever you see very a peculiar molecular weight, higher molecular weight for the specific proteins, those lines can be isolated, and those particular regions can be eluted and go for subsequent genomic analysis. And this is one of the studies done at SRS Melalatur at TNAU. And now that what I want to register here is this COG, CO Gudiatham 6 has shown higher cane yield. You can see this is when we have compared the same line with very popular variety 86032. So it has given higher cane yield as well as CCS percent. So this is also a flowering type. So those who are interested in developing drought tolerant varieties, you can use this COG6 also, one of the parents. And again, somoclones, creating somoclones is very easy in sugarcane. We can try the same method for developing salt tolerant sugarcane varieties. So to my knowledge, there is no specific salt tolerant somoclone variety grown in India, but anyway in other countries they very regularly use this technique for developing salt tolerant sugarcane varieties. It works out well for rice and other crops also. So the whatever somoclones they have developed through this tissue culture, they show high level of calcium content in shoot and high level of sodium potassium ratio. So these two are the major parameters that confers sodium. So they have to be very strict in excluding the sodium, whatever salt tolerant line. So they have tested the somoclones for these two ion level. So and those lines have shown promise for calcium level as well as for high potassium level. And there are some of the genes, that is sugarcane drought tolerant gene 1 and 2 are there, but one I could see in one of the literature very peculiar, this SCDR1, even though it shows discriminating drought tolerance level when it is transferred to other crops, within the sugarcane it is not showing that much discriminating ability, but SCDR2 is better than SCDR1. And again, for drought tolerance, sugarcane dirigent protein, SCDIR, and cold stress also, there are some genes, upregulated genes they have reported. This is from Brassica. They have used for transgenesis also. This is Brassica gene and drought tolerant dehydrin protein and other genes. So there are many, even for waterlogging, there is one gene. So using this latest technique CRISPR, they are trying this manipulating this particular gene SCAPX6 and LTP. So which is connected with the methyl jasmonate or abscisic acid. So coming to gene silencing also, this MIR, so this is one of the very handy technique. Now it is followed by many of the biotechnologists. So this is gene at the expression level using this microRNA interference, they are able to obtain good lines with sugarcane lines or clones with cold or salt or drought tolerance. This MIR 164, 394 are for drought and 159 for salinity. Coming to waterlogging, so waterlogging is major problem, especially in northeastern states and also in Bihar. So this is also of great concern because it gives yield reduction as well as sugar recovery is also affected because of this waterlogging. Of course, we are fortunate that we have some good varieties as well as donors for this waterlogging. Co 951, 975 and Co 62, 175. And BO3 is completely resistant for this waterlogging, which had maximum root density, so that it is able to provide good oxygen even under waterlogging condition. So this is the anatomical. So you can see the in the tolerant lines this parenchyma formation, which helps in coping up with the waterlogged condition, and also high phenol oxidase activity. And you can see the adventitious roots, stint root arising from the top nodes also, and less increase in ADH activity. So these are all the parameters to screen the clones for waterlogging conditions. So the genes alcohol dehydrogenase and submergence genes were expressed in Co62175. So compares 86032 is sensitive to waterlogging, whereas the 62175 is tolerant. So in this particular tolerant variety, all these three genes have expressed very well. So this can be a dependable one. So of course, either using the biomarkers or with the DNA markers, we can screen the varieties. So this I can skip. So there are very few QTLs have been reported. Whatever QTLs which I could find from the literature is only for ethylene pathway, which is playing a major role, especially for the abiotic stress. And we have good number of genetic stocks for abiotic stress, and almost all the sugarcane breeders are aware of this. So in SBI portal also it is available. So many lines are there. So we have to carefully choose the parent, considering other aspects like red rot resistance and other things, and going for hybridization. And there are some multiple abiotic tolerant lines we have with the help of the great efforts of the breeders. They have given for given varieties or sugarcane varieties for more number of abiotic stresses, like I have listed out here. So drought, waterlogging, salinity. So they have combined many abiotic stress tolerance mechanism in a single variety. And these are all the drought tolerant varieties from TNAU 25, SC6, and this SI. See, we have one research station at Sirigamani, Trichy district of Tamil Nadu. So that is exclusively for screening the varieties for early drought as well as for late waterlogging conditions. So and that particular center is situated in Kaveri Delta zone. Early stages, we experience a drought, so we can screen the varieties naturally. So whatever varieties coming out of that particular research stations are having the potential of early drought tolerance as well as late waterlogging. And again, Koji 6, which I had mentioned earlier, this is tolerant for drought, salinity, as well as for tannary effluence. So, in Vellore district of Tamil Nadu, we have the problem of tannary effluence in the irrigated water. So, this particular variety has been identified to be tolerating that heavy metal concentration in the tannary effluence also. So variety is tolerant for drought and salinity. So say now that we have gone for the concept of straight cane to enable mechanization in harvesting. So very good pictures of all these varieties you can see. And these are all the multiple abiotic stress. Almost all the research stations are working on drought tolerance. And there is these are all the hybrids which I have listed in earlier slides were through the single, that is biparental mating. Now what we suggest is polycross also, that would give you more favorable genes. So with that aspect, so some of the polycross material have been developed using this COC 772, which was subjected for this polycross. With that they have come out with CO 06027 for drought and 22 and 27 for salinity. And this particular 007 and 020 has been reported to be tolerant for flooding and waterlogging. That's what, instead of biparent, as far as this abiotic stress is concerned, already sugarcane genome is a complex one. And again, the mechanism underlying this abiotic stresses are again a complex. So many genes are involved. So if at all we want to have a abiotic stress tolerant varieties, it is better to augment as far as possible, as many number of favorable genes. So that could be done with the help of polycross. And coming to the biotic stress, I will be concentrating on red rot smut and borers. So these are all the biochemical bases. I think you are well aware. This is phenol production. Is high phenol producing clones usually will be showing high level of resistance for biotic stresses. And so much of work on molecular basis has also been done. They have identified the expressing gene tag. They have tagged the genes based on the expression level also. And usually now that pathologists have come out with a very good technique of CCT method, controlled condition, they are screening the clones for red rot resistance. And there is, of course, there is one old technique called seedling blasting technology, which we have tried in TNAU. Just we have tried this technique with GC fluff, seedling, and we have come out with only 13 seedlings. When we just spray the spores, as per the specification given by the pathologist, we could realize only 13 seedlings. But to our surprise, all the 13 clones, which had survived this high level of blasting during seedling stage, have been found to be with spontaneum type. So cane forming clone, I could find only one. Yes, still this work is going on. At least we can identify the strong donor. We can be confident enough with the red rot level resistance that can be used as the donor. And of course, a role of transcription factors in red rot resistance is repeatedly reported in all the literature. So the candidate genes, transcription specific and secondary metabolites, proteases, transporters, and peptidase have been reported. That is this Colletotrichum falciparum, which is the causing pathogen for this red rot. So Coimbatore falciparum transcripts encode a large number of secondary metabolites and membrane transporters. So to my surprise, they have reported that is 13,320 genes are involved in this metabolism related to the defensive mechanism for this Coimbatore falciparum. Again, for SMUT, this is so many phenolics are involved, major genes also, they have catalase genes they have identified. But in one of the report, interesting to me is what they claim is non-destructive method. Even at the level of bud, we need not wait for the whiptail development for the SMUT screening. Even at the bud level, you can screen the SMUT resistance with the help of NIR, near infrared spectroscopy. So breeders should be well conversant with screening, even though physiologists or pathologists or entomologists will help us in screening, but we are also supposed to learn or to understand or should be confident enough to screen the genotypes. And disease resistant genes, especially for viral diseases, the specific genes have been reported, and DNA markers are also available. So for example, this sugarcane grass resistant, these two are the resistant types, and we have the EST based markers also. And for brown dress, which is not that much prevalent in India, we have some good number of molecular markers. So in vitro selection is possible. We have also taken up this work at TNAU, because using this pathogen culture filtrate, we can screen the callus as well as the regenerated plants under in vitro condition, and later on we can take it to the field. So sugarcane somoclones regenerated through the callus of COS 91279 showed wide variation for red rot resistance. Again, 4 isolates. They are just mixing all the isolates to ensure the resistance level. So this particular line, which has been developed through somoclonal variation, has shown promise for red rot resistance. So what we have observed with the preliminary studies, these are all the COC somoclonal variation. Even we could see some of the lines under field condition. We could see the variation for mealybug incidence and for the red rot resistance. Even though many of the somoclones were showing susceptibility for red rot, even within that susceptibility, we could see the wide range of susceptibility level. Even it has got the score of 5 or 7. Within that 7 level, we could see the spread of this pathogen. Okay. And inheritance pattern of coming to the insect resistance. So we are fortunate breeders, so it has strong additive genetic variance is there for the insect resistance. But fortunately or unfortunately, we don't have a strong donor for conferring this borer resistance. So traits associated with borer resistance, that is morphologically, we can screen the clones with based on leaf surface with high silicon content, rind hardness, leaf sheath tightness. So that will contain the insect entry or the larval entry, and high fiber content. Of course, we have to take care of this fiber content. We have to optimize the fiber content, and stalk pith as well as plant vigor. So diameter, if it is more, less incidence of pest is there. Besides this morphological characters, I could see, or we have one good report that even antibiosis, which contains the growth of the larval. So they have reported 2 lines, HO 08901 and CP. I think this is the only report I could see as far as this antibiosis is concerned. And donors, we have good number of donors for diseases like Fissionarum itself is a good donor for SMUT, and Spontaneum I could see. I was so excited in the previous slides. I was discussing about that blasting product. Even in the seedling, we have gone for the blasting the spores and come out with some of the clones. Again, it has confirmed that Spontaneum. So whatever clones I could see in the field with the resistance level, which has got surveyed, were of Spontaneum. So Spontaneum, thoroughly it is a red rot resistant one. We have to still exploit that, and we have to go mine this at a genomic level and identify the particular genomic, selective genomic region that is responsible for red rot. We need to have this elaborate studies on this spontaneum. And robustum is also good for the waterlogging as well as for some of the diseases. And you could see very Naranga only shows this pest resistance, that too not for this borers, and Erianthus nematodes resistance it is giving. So again, one another interesting thing is sweet sorghum. While I can't say it is a wild relative intergeneric cross, that has given good a level of resistance for a red rot. So this is GU08SS66. It is one of the genetic stocks which you can.
Namaste, ma'am.
Hello, red rot resistance. And another work by Alamelu from Sugarcane Breeding Institute, they have used this robustum for introgressing red rot resistance as well as drought resistance. I think I'm taking more time. I'll just rush up. And microgene licensing is also silencing is also used for disease resistance. And polyketide synthase 1 gene in coletum, that is red rot causing pathogen, has been applied. This particular technology is technique has been applied to control the activity of this gene. And expression of sugarcane mosaic virus coat protein gene was downregulated by applying RNAi technology. Again from SBI, Dr. Vishwanathan has validated the targets of sugarcane streak mosaic virus encoded miRNA in sugarcane. And there is much collinearity between this sugarcane and sorghum bicolor genome for identifying putative. So that might help in identifying putative candidate gene that could have a role in plant virus interaction. So genetic transformation has been done very well for sugarcane, but you could see many diseases have been taken care of: Fiji disease, red rot, and mosaic virus using this chitinase gene or coat protein gene. And for insect resistance also, both this Cry1AB and GNA have been, Cry gene have been used for developing sugarcane varieties with borer resistance. And what they report is foreign Cry1AC gene integration and endogenous borer stress related genes have a synergistic effect. So this we have not seen in cotton or in maize, but this is in sugarcane because it is a aneuploid and complex genome. What they have reported, this has a synergistic effect with the already existing defensive insect defensive genes in sugarcane. So suggested modification. So see what we have experienced is, though we have got to the level of higher cane yield and sugar recovery, but there is a stagnation. Even though we have succeeded in developing resistant lines for red rot or other diseases, there is a stagnation in sugar recovery. So for that, I'm just proposing, so we can, while selecting, so far we had been concentrating on choosing the parents based on their combining ability. Now that we have to directly develop a pre-breeding material using this wild relatives also. And wherever the stress, abiotic stress or biotic stress is a major problem, we have to concentrate on polycross. Already developed polycross material, pre-breeding material or genetic stock should be widely used. And selection of genotype types not only based on stock population or cane diameter. We have to take care of this pest and abiotic stress tolerance, and also stay green. This is a easily identifiable trait. So stay green types hold good for drought tolerance, salinity tolerance, as well as for water logging. And ground nursery, not only for bricks. So only for bricks we can select because it has got high heritability. Other characters we can have lesser selection intensity. And see, even from starting from clonal nursery, we have to take care of screening for biotic or abiotic stresses raised in augmented block design. So even though population improvement is helpful for augmenting many favorable genes, it is not very, it cannot be applied much in sugarcane because it might cause inbreeding depression, at least to the level of S1 cycle, recurrent selection, S1 cycle we can attempt, so that the genetic, that is, we can just widen the genetic variation in this. And this is the one what I want to register here is you have to take care of this ratoon crop of the ground nursery or clonal nursery. Wherever it is possible, we can subject those ratoon crops for the very important or very problematic stresses like a drought or red rot. Wherever it is possible, we need not leave this ratoon crop of, especially of the clonal nursery, and we can make use of that ratoon crop for screening, so that the time requirement for screening for all the pest and disease can be fringed. So that what I want to register here is going for this ratoon screening also. And in the ground nursery, we can give only 25, see, unless there is good sugar recovery. So the first screening, let it be for the high sugar recovery 25%. In the subsequent generations, we can concentrate on cane weight like 1 kg or 1.25 kg. So at this level, clonal nursery, we can, whatever marker is available, no, as far as we dehydrin is there, some resistant gene analogs are there. So miRNA related markers, DNA markers are available. So wherever it is available, so those markers could be made use of at the level of clonal nursery and shortlist the number. And immediately after screening this, we can take it for the CCT, rapid screening for red rot and other things. So what I would suggest is, so when there is a very dire need, we have to develop drought, salinity, within a short time we can practice this method, and we can go for the micro propagation even at the level of clonal nursery. Simultaneously you can go for the row yield. So instead of 4 rows, we can just try for the 2 rows in that. So this I want to mention or register here is to reduce the time for the development of multiple resistant clonal development or clones development or variety development. So similarly, as we do for rice, pyramiding of gene is very essential. And of course, there are some challenges. Genomic selection helps in capturing the non-additive gene effect. So in maize, so when they have tried this 2 cycles of phenotypic selection, this is the phenotype. But when they went for this one phenotypic selection followed by one genomic selection, they could get good results. You can see the result here. So in sugarcane, now that genome has sequencing is also in good progress, and mosaic monoploid genome monoploid reference sequence is also available for studies. And magic studies which we have achieved in rice, the same can be practiced in sugarcane. So we can involve the all the saccharum complex pre-breeding material with like barberry, sinens, robustum. So all can be used in a polycross, or 12 crosses or 18 crosses can be made. So which would help in developing a good pre-breeding material with the augmentation of more number of favorable alleles for this biotic and abiotic. And Jivas, so Yang et al. From China, Thailand, I suppose, they have applied this Jivas and come out with the yellow leaf virus disease resistant line. So this Jivas would help in identifying the candidate genes also. So at the last, I would request the youngsters especially to go for the speed breeding, even though it's in the infantry stage. So we have to alter the photoperiod length and reduce the breeding cycle. That is, you have to facilitate flowering even at the stage of 5 months old crop or 4 months old crop for the crossing, so that we can reduce the breeding cycle time. In all crops, in chickpea at Ikrisite, they have succeeded in developing varieties using this speed breeding. So within 7, they have achieved 7 generations per year. So hardly hitherto they had been practicing 2 generations. With the help of the speed breeding, they had gone for 7 generations. That had helped him a lot in reducing the breeding cycle. And a way forward and conclusion. So more investment on development of biotic and abiotic research investment should be there. sugarcane varieties, especially, and we have to concentrate more on drought tolerance and widening genetic bases through population improvement, related genera in progress line, and magic population, and candidate gene based gene marker development. So many candidate genes have been reported, especially for drought and salinity, but no tightly linked markers, DNA markers have been reported. So much explosive study on identification of DNA markers is required. Gene pyramiding is also another task we have to take care. Like 86032 is a very good variety, but it is still drought sensitive. So we can take that variety and try to introgress all the favorable alleles there. And utilization of ratoon crop for clonal nursery. So we can exploit the ratoon crop for screening, simultaneous screening for pest or disease resistant, whatever may be, according to that region in which it has been grown. And especially the capacity building on speed breeding is very much required. That should be given more emphasis, and encouraging shuttle breeding also. As I said, so drought or salinity it will not occur throughout the year, and the occurrence of drought and the level of nature of drought also varies. So to confirm or ensure the drought tolerance, we can go in for multi-location like shuttle breeding, for which we should have a good network program. Thank you very much for patient hearing.
Thank you. Thanks a lot, ma'am, for such valuable presentation. Uh, Now, if we can concentrate on chat, ma'am, there are a few questions, I guess.
Yeah.
Sugarcane wilt resistant genes, question regarding that. Another was what GWAS stands. Can we ask questions directly to ma'am using your mic? You could directly ask questions. Ma'am, the chat window is
Yeah,
in
yeah.
very
From
formative.
the chat, any sugarcane wilt resistant genes. So, with my knowledge, I could not find any report on wilt resistant genes. And GWAS is genome wide association studies. So you have to raise the even the germplasm or the genetic resources across locations and go in for the studies.
Any other question or query which we want to direct to ma'am, please?
Madam, can you elaborate the gene silencing concept?
Yeah, gene silencing is it's a kind of expression. See, suppose there is a positive regulator that facilitates the pest disease, that is the pathogen entry or its activity on the plant. So that can be curtailed, that can be silenced using this microRNA. So there are specific genes for this microRNA also. We have to identify those genes and activate that. So this can be done with CRISPR also.
Okay, okay. Thank you.
So actually that microRNA cleaves, that particular, after this transcript is produced for that particular activity to encourage the pathogen, this microRNA cleaves that transcript so that its activity is stopped.
Any other question, please? I think we are done with the questions, and we are just in time for the this presentation to conclude. Thank you from the organizers, participants, and NADCL. Thank you, ma'am, for such a lovely presentation, and thanks for being with us. Thank you, ma'am.
Thank you so much for giving the opportunity. Thank you, one and all.
Participants can leave and join back at 2:30.